So we wanted to look at pharmacokinetic parameters, really, of mitragynine and how much 7-hydroxymitragynine is being produced after a single dose, or either orally or IV to Sprague-Dawley rats. In this case, these were all female rats. We don't see much of a sex difference in the pharmacokinetics. We do see some sex differences between species, but I don't have time to go into that today either. The bottom line here is the take-home message. Excuse me, orally, we convert about 9% of the mitragynine dose to 7-hydroxy versus IV, there's only about a 3.5% conversion to 7-hydroxy. We also found that 7-hydroxymitragynine is totally unstable in human plasma. So we were doing plasma stability studies because we wanted to do a lot of studies with 7-hydroxymitragynine in animals. We were doing cross-species plasma stability studies. And this actually, we found that mitragynine is converted in the liver to 7-hydroxymitragynine, which we already know and we showed on the last slide. But in plasma, 7-hydroxymitragynine actually undergoes this semi-pinnacle rearrangement. It is protein driven. We don't know what protein does it yet, but this produces mitragynine pseudoindoxyl, which has never been detected from the plant before. And again, it's not from the plant. This is a metabolite that humans are even forming, and it seems to be a human-specific metabolite. So if you look at the insert of the graph on the right, we were actually seeing this metabolite present by tremendous amounts versus mouse, rat, dog, or monkey, tremendous amounts in humans. I actually made the lab scale up this reaction so we could isolate enough and perform NMR. And that's where we came, because the mass was identical to 7-hydroxymitragynine for mitragynine pseudoindoxyl, but the retention times changed. So we knew there was something there. Anyway, they ended up publishing this in ACS Pharmacology and Translational Science and got us a little cover feature. And this has been interesting work, which led us to more questions. So what's going on when we give mitragynine orally, and how much 7-hydroxy are we producing, and what's present in the plasma versus the brain? And so we took the ED50 from some of our analgesia studies in mice, and we dosed those mice, and we collected plasma and brain at various time points, and we dosed them with mitragynine, not 7-hydroxy, but what we wanted to see was how much 7-hydroxy was present in those animals. And you can see that the brain to plasma ratio of mitragynine itself is about 2.4, and the brain to plasma ratio of 7-hydroxymitragynine that's been metabolized or produced from that mitragynine dose is about 1.65. So there's more 7-hydroxy in the brain than in the plasma. There's more mitragynine in the brain than in the plasma. I think what really surprised us is that we took this and then decided to look at the ED50 of 7-hydroxymitragynine orally, and we saw the reverse. So 7-hydroxymitragynine doesn't seem to cross the blood-brain barrier as efficiently. The brain to plasma ratio is less than 1. So there's more in the plasma than in the brain. And we knew that the metabolite could be generated, this mitragynine pseudoindoxyl, albeit in low amounts. We still see it in the mouse, and we saw that have the same issue. More of it's in the plasma than in the brain. In fact, it's an extremely low ratio. And what we believe that's going on there, when we start doing a little bit of pharmacokinetic modeling, we looked at mitragynine's concentration versus its effect, and so we could calculate the log brain concentration over the log maximal percentage effect, and we could generate a range of pharmacological response, and that mapped really nicely onto our actual observed values. But what's more important is when we looked at the efficacy of 7-hydroxymitragynine versus its effect, you can see that in the blue with the red, and the red are the observed. What was reassuring to us is that the maximum 7-hydroxy concentration that can be generated after mitragynine administration is far below where the efficacy could be found. So this was just published in February. This is some of the hottest new data. This is refuting some work that's come out saying that 7-hydroxymitragynine is responsible for kratom's analgesic effects or mitragynine's analgesic effects. And we don't believe that to be the case. We don't believe the amount that's generated through metabolism is pharmacologically relevant. There are some questions as to why mitragynine, when mitragynine is given by itself, you see higher brain to plasma ratios than when 7-hydroxy is given by itself. But we've been dealing with those, and I'll be happy to answer any questions around those. I would just jumping back on the cytochrome P450s. I know we're getting close to time, and I want to jump ahead. CYP3A4 seems to have the predominant role here. The clearance of mitragynine is really mediated by CYP3A4, and 7-hydroxymitragynine is a resultant from that. We have seen a total of 12 metabolites in microsome studies and 31 metabolites in hepatocytes, and those are human liver microsomes and hepatocytes. We also wanted to look at CYP inhibition, and I haven't talked about any of these other alkaloids yet, so I'm going to go back here. And what we saw was really that there's not any significant inhibition of cytochrome P450s, but we do see moderate inhibition of CYP2D6 with mitragynine, the major alkaloid. And this could be somewhat responsible for why we've seen some adverse events with other CNS drugs that are metabolized and cleared through CYP2D6. We haven't done any detailed studies on that to prove that there are any issues or are any recommendations to avoid those things. For the most part, many of the other alkaloids within kratom don't have issues either.